Sorry about being late. There's been a lot of trouble for my logging in. I wasn't able to get in with the link, Zoom link in Canvas. I had to log in at, zoom.com and I didn't remember my settings on that, so I had to go look up my passwords. And then it had me verify with what is it, a code they sent my phone. Anyways, are there any questions before we begin? All right, I'm not hearing anything. I'm going to share my screen, although I better shut that down before I share my screen. Let's see. All right, you should be able to see my screen now. Can I have somebody verify you can hear me?
We can hear you.
Great. All right, today is October 14th, and we should be doing Lab 5. Let me inform you that you should read about the unknown project instructions. There's a file on Canvas. You should have that read by next week, and let me show you where that is. This is why I was trying to get into Zoom from my Canvas webpage and it wouldn't work. Let's go here. No, I'm not sure if that's Zoom or not. Oh, here it is, unauthorized. Let me, it looks like I accidentally logged out of my, Canvas website. So let me log in again. Hopefully I won't have to put in any information. No, I don't. Sorry, it was in here. My computer seems to be a little slow today, or else the network is really slow. All right, so scroll down here to week 5 lab, and then the unknown instructions, which hopefully you can see that right there. That's the file I want you to read. It's about the instructions about the unknown project. Any question about that? All right. Um, so let's begin lab 5. Guess I don't need this, but we'll keep it up. Can get it in time. All right, lab 5, cultivation of bacteria, talking about culture media, inoculation, and isolation. There is some reading before the lab, but not very much. Read about culture media on page 157 to 162, and then read about obtaining pure cultures on page 162 to 163. There are some video clips in today's lab. There is no virtual lab. We used to have one, but it used Flash, and Flash is no longer supported, so it no longer works on computers. Upon successful completion of this lab, you should be able to, one, understand the importance of proper specimen collection and transport to the lab, two, explain the 5 principal steps used by microbiologists to process specimens, three, differentiate between the various types of culture media, four, explain the purpose of the streak plate culture technique, that is, streaking for colony isolation. And then 5, defining the following terms. What's selective media, differential media, and reducing media? Any question about any of these objectives? All right. The ability to isolate pathogens from clinical specimens submitted for culture is only as good as the collection technique and timely transport of the specimen to the lab. Some microbes are sensitive to desiccation, meaning they dry out, and some are sensitive to the exposure to oxygen, most anaerobes. And therefore, these cultures require a special transport medium. And in fact, all living cultures require a special transport medium. Once a specimen is received in the lab for microbiological culture, microbiologists employ the 5 principal I steps. I don't know why that is like that. It looks like it's on the box of that. Anyways, the 5 principal I steps are 1, inoculation, 2, isolation, 3, incubation, 4, inspection, and 5, identification. Inoculation is just placing the specimen on an artificial culture medium to initiate its growth. Isolation is we have to spatially separate microbial growth into distinct colonies. And in fact, when we study it, we'll have to make sure we have a pure culture that we obtain from passing the culture usually through 2 colony isolation events. And then 3, incubation, placing the culture medium in an environmental conditions to facilitate its growth. We normally incubate our culture in an incubator, and I'll show you that in a minute. 4, inspection. After the culture is then grown up, you then examine the culture for the presence of microbial growth. And then 5, identification. Usually you have to perform specific tests to identify any pathogenic microbes that have been isolated. It used to be, probably when I was a kid, maybe even when I was in college, if they grew a culture from your throat on a blood agar plate and you got a small colony with hemolysis around it, they automatically assume that it was Streptococcus pyogenes, which most of the cases it probably was. But in reality, it could be something else, and you should run other tests, but that's certainly a major one. And then with strep throat, you only have to run at most 2 other tests besides growing it on blood agar and getting the hemolytic activity to a small white colony on the plate. Anyways, these 5 steps will be described after we discuss the different types of culture media. We have different types of culture media. You should be already familiar with general purpose media, such as Na, nutrient agar, Na, that's nutrient agar with yeast extract, and TSA or tryptic soy agar. We discussed these general purpose media in Lab 1, and they were just a rich media to grow many types of organisms, but they did not grow everything. Specifically, they would not grow a fastidious microbe. A fastidious microbe requires a very specific nutritional additive before they will grow. And most fastidious microbes, we grow them on an enriched general purpose media, usually just called an enriched media, but you could call it actually a enriched general purpose media. For example, you can add 5% sheep's blood to TSA, and then we have blood agar, which will allow Streptococcus pyogenes to grow, a fastidious organism that requires specific nutrients to be supplied, and the blood supplies the nutrients for Streptococcus pyogenes, the bacteria that causes strep throat. The blood requires the nutrients for Streptococcus pyogenes to grow. Another enriched general purpose media is chocolate agar. It's the same as blood agar. It's just the blood is added immediately when the agar comes out of the autoclave. So the agar is very hot, and that lyses the red blood cells, creating a brown color that reminded someone of chocolate, and that's why it's called chocolate agar. There is no chocolate added to the agar. It's just blood cells that have been added when the agar is hot, and that causes the blood cells to lyse, giving the color a brown color. Any questions about enriched general purpose media? All right, we also have selective media, and these are media that contain chemicals that inhibit or halt the growth of some organism, but allow other organisms to grow. Some things to keep in mind about selective media. First, selective media isn't necessarily perfectly selective, meaning given enough time, organisms which are inhibited may grow on the plate. And second, even the organisms that are supposed to grow on the plate, they grow more slowly or form smaller colonies than they would on a general purpose media plate. There's many kinds of selective media, and we'll talk about a few in this lab. And then there's differential media. These are media that contain chemicals that produce visible differences among different groups of organisms when they're grown on that same media. These differences are unrelated to how well the organism can grow on the media. It's just that the colonies will appear different. Usually, the differences are visually distinguished from each other. And lastly, there's reducing media. This is media to promote the growth of anaerobic bacteria because they require low or no oxygen conditions in order to culture the organisms. It's important to note that a culture media can be classified into more than one category. For example, McConkey's agar is both selective and differential. And then blood agar is both differential and an enriched general purpose media. So here's McConkey's agar. On McConkey's agar, if you get growth, then it's likely to be a gram-negative growing because McConkey's agar has chemicals that select against the gram-positive bacteria. You don't need to know the chemicals, but actually, let's see if I can remember. One of them is um, crystal violet is in the media, and that selects against gram-positive cells because the crystal violet gets stuck in their cell walls and doesn't allow it to grow. And then it has bile salts in it, which also select against gram-positives. Many gram-negatives can grow in bile salts because the gram-negatives tend to grow in your intestines, and you produce bile salt to help you digest certain foods. Anyways, that's why it's selective. You don't need to know the chemicals, but you should know that McConkey's is selective and selects for gram-negative. So if you get growth on the plate, then it is likely to be a gram-negative organism. And it's also differential, and that is if the bacteria can ferment the sugar lactose, you'll see pink colonies like this, or even pink around the growth. Like right there, the media in between the streaks is pink, and that's because of lactose being fermented. You don't need to know this, but we'll just mention it anyways. A lactose is fermented that lowers the pH, changing the color to pink, meaning the pH indicator turns pink at this pH. And if the bacteria can grow on the media, it's gram-negative, but if it cannot ferment the sugar lactose, the media will be its normal, the media and the agar or the growth will be its normal color. So this bacteria growing on the media, McConkey's media, is off-white in color. And it depends on what the bacteria is for what color it'll be, um, but it'll be its normal color. Any question about any of that? It will not be pink. It will be its normal color, whatever color the bacteria normally is. So McConkey's is both selective and differential. Blood agar is both an enriched general purpose media, meaning it'll allow you to grow fastidious organisms that require whatever additives blood provides. And for Streptococcus pyogenes, I might actually remember the additive. One of them's a B vitamin, like B6 or B12, something like that. And the other one, I don't remember. It might be nicotinamide. So that's why it's an enriched media. The blood provide nutrients that the organism, if it's fastidious, may require. And different fastidious organisms have different additives that need to be added to the media. And then blood agar is also differential. It allows you to differentiate between the hemolytic activity of the different bacterial species. Like this bacteria species has no hemolytic activity, and that's called gamma. That's actually the lower Greek letter for gamma right there. And there's no hemolysis. You can see the red blood cells going all the way up to the bacteria. And then there's alpha hemolysis. That is where the bacteria are partially lysed, or the red blood cells are partially lysed by the bacteria. And let me blow this up a little bit so you can see it a little better, if I can get to it. I've got that stupid icon from, oh, come on. Grammarly there. All right. You'll notice the darkening before the bacteria, which is the alpha and the off-white. That darkening, which may appear greenish, but oftentimes it's dark like this. And you can see it inside the alpha. It's totally dark, darker than the red there. And so it's red, dark, I don't know, dark red. And then we have the off-white of the bacteria. And then inside the alpha, it's dark red again. And that darkening is from alpha hemolysis, the partial hemolysis of the red blood cells. That differs from beta hemolysis, which is the complete hemolysis of the red blood cells. And you can see the white beta there, that being the bacteria streak. And then this clear zone around the bacteria, as well as inside the beta, is from the complete hemolysis of the red blood cells. You can actually look through the media where there's beta hemolysis or complete hemolysis. And I like to shine it up to the light to see the light through it, or you can shine it up to the clock and determine what time it is. It's up to you what you want to do. But the point is, I like to look through it to make sure it's beta hemolysis. And you might notice a darkening ring around the beta hemolysis, and that's normal to see a ring of alpha hemolysis around the beta hemolysis. So beta hemolysis, alpha hemolysis, and gamma hemolysis, that is why blood agar is a differential media. Any questions about McConkey's or blood agar? Let me shrink this down now. Let's go there. All right, this table is showing you the different types of media. General purpose media, there's three we've talked about: tryptic soy agar, TSA, nutrient agar, or Na, nutrient agar with yeast extracts, and that's Na. And then there's a differential media and an enriched general purpose medium, and that is TSA with 5% sheep's red blood cells added, and this we call blood agar, or BAP. and it's a differential and enriched general purpose media. Then we have CAP, which is only an enriched general purpose media. Then we have McConkey's MAC, eosine methylene B, or excuse me, eosine methylene blue, or EMB, or hectone agar, or HE agar. These three are all selective and differential. All three of them are selective for gram negatives, so gram negatives will grow on these three media. And they're also are all three of them differential on the basis of the fermentation of lactose. McConkey's I've already talked about. If it can ferment the sugar lactose, it will turn pinkish color if it's a gram negative, because it's gram positive. You cannot, let me add with the selective and differential media, you cannot state the differential ability of this media if the bacteria will not grow on the plate. Okay, so if you get no growth on a selective and differential media, you cannot state what the differential ability of the cells are. You have to have some growth in order to determine the differential ability. Anyways, they're all differential on the ability to ferment lactose. McConkey's will be pink if it can ferment the sugar lactose. EMB, if it can ferment the sugar lactose, the media will either turn dark, almost a black color, or it can turn a metallic green color if it ferments the sugar lactose. And then hectone and enteric agar, if it can ferment the sugar lactose, it will turn a salmon color. Salmon is a yellow-orange color. Then we have phenyl ethyl alcohol, or PEA media. It is selective media, and it selects for gram-positive bacteria. So if you want to grow gram-positive bacteria, you put it on PEA. If you want to grow gram-negative bacteria, you put it on MAC, EMB, or HE agar. And then we have mannitol salt agar, or MSA, and it is a selective and differential media. It is selective in that only salt-tolerant organisms can grow on mannitol salt agar. It has 7% sodium chloride in it, and most organisms will not grow in 7% sodium chloride. In fact, most organisms require 2% sodium chloride or less to grow, and MSA, or mannitol salt agar, has 7% sodium chloride. Staphylococcus species can grow on this plate, as can, I think, Enterococcus. There is 1 gram negative. Let me see if I can remember it. I think it's Enterococcus aerogenes can grow on mannitol salt agar. Or maybe it's, no, it's Enterococcus cloacaceae can grow on mannitol salt agar. But there are a few gram negatives that can grow on it. So if it can grow on it, let me warn you, it's more than just Staphylococcus. Staphylococcus, all members can grow on mannitol salt agar, but there are many other gram positives and a few gram negatives which can grow on mannitol salt agar. Mannitol salt agar is differential on the ability of the bacteria to ferment the sugar mannitol. If the bacteria can ferment the sugar mannitol, the colonies will be yellow, and you'll see a yellow halo around the colony. And you should really go by the yellow halo, because if you were growing Micrococcus luteus, for example, on mannitol salt agar, it can grow on mannitol salt agar, and Micrococcus luteus itself is already yellow. And then Staphylococcus aureus, if it were to grow on the mannitol salt agar, it has a yellow colony too. So you should look for the yellow halo in the media around the colony to determine whether it is capable of fermenting the sugar mannitol. An easy way to remember what's in the mannitol salt agar, it has mannitol in it and salt, sodium chloride. Any question about the different type of media? All right, inoculation is just putting the bacteria on the media to begin the growth. Obviously, you have to put it in the incubator. Isolation, you're going to have to get a pure culture because if you have a mixed culture, which you would always get from getting a clinical specimen, you need to isolate different colonies and get pure cultures to find the colony of interest which is causing the patient's symptoms. And usually to get a pure culture, we'll streak out a colony. So that's one isolated colony, although that's not a streak, but this is an isolated colony. You can take from this isolated colony, put it on a plate, streak it out for isolated colonies, and then you take cells that have gone through two colony isolation events, this one and on this plate here, to get up a culture that we'll call probably a pure culture. Even then, it's only about 95% or greater chance of it being a pure culture. In microbiology, we never get 100% chance that you'll get a pure culture. Any question about any of that? In fact, the current chair of the biology department was trying to study some organisms that were let's see, fixing nitrogen from the air, and he was trying to get a pure culture, and he kept streaking it out, and he couldn't get a pure culture of the culture he was wanting. It turned out he needed to have two organisms on the plate in order for him to get the culture that he wanted, meaning he had to have two species growing, and one pulled down nitrogen from the air, the other changed that form of nitrogen to another form, and then the two species together could grow on the plate. One species itself could not grow on the plate. And so he never got a pure culture, at least from streaking. I'm not sure what he did to, he probably had to grow the bacteria on another media supplying nitrogen before he got his pure culture. Louis Pasteur was the first to develop the concept of pure culture. He had a very difficult way of getting a pure culture, and that is he would make different dilutions of his mixed culture of bacteria, and then he would make 10 copies of each dilution. And if ever he got one dilution where 9 out of the 10 tubes did not have any growth, and one tube had growth in it, he correctly surmised that the one tube with growth was a pure culture, meaning a high chance of it. Edward Koch or Dr. Koch had a much easier way of obtaining a pure culture. And what he did was he streaked a mixed culture of bacteria onto a solid media. He got an isolated colony, took that cells from that colony, streaked it out on a second colony, and then grabbed an isolated colony from the second plate to set up his pure culture. And like I said, even then, he's not guaranteed of a pure culture, but it's greater than 95% chance that you'll get a pure culture. And that's good enough in microbiology. Oh, there I've got talking about Robert Koch and how he obtained his pure cultures by streaking for colony isolation. And remember, you have to streak it out 2 times. On the first plate, take from one isolated colony and then streak it out on a second plate. And then when you take from an isolated colony, you can set up the pure culture. So how you streak on a plate to get isolated colonies is you take your mixed culture, streak it on quadrant 1, and you should do it about 20 to 30 times. We're not seeing the entire streaking here. And then you sterilize the loop, and then you put it on quadrant 2 in this area here. You don't start where the bacteria is, and then bring it back into the low region of quadrant 1. Let me blow this up so I can show it a little better. So what you should be doing is cooling the loop after you sterilize it on quadrant 2 right here, and then you streak it into the low portion of quadrant 1, meaning you shouldn't come into this region. This is a mistake here. You shouldn't come into this region because the bacteria are going to be most concentrated on this streak, and as you streak further down, the bacteria numbers will decrease. So the lowest concentration of bacteria will be here, and the highest concentration will be here. So you don't want to get in the highest region. You should be in the lower regions. So you should be coming like here. And you should go, at least 2 times, maybe 3 or 4 times into the lower region of quadrant 1 to make sure you get bacteria to streak it into quadrant 2. And there we have quadrant 2. And then you take the loop and you sterilize it by putting it in the flame or the bacterial incinerator, getting it red hot, and then you cool it down on quadrant 3. And this is actually good. They've started now on quadrant 3, and there won't be any growth here because you don't have any bacteria on your loop. And then you come in the low region of quadrant 2. So you should really only come about this far. You shouldn't come into this region because the bacteria will be most concentrated on that streak, and then most concentration on that streak, and then it'll be diluted in this streak. So you should come where it's diluted. So you come 3 or 4 times into this region to pick up cells, and then streak out on quadrant 3. And then you sterilize your loop, cool it on quadrant 4. And this one they've actually done well, and that is you only come in the dilute regions of quadrant 3 to pick up the bacteria, and you come into quadrant 3 about 3 or 4 times to pick up cells from quadrant 3 and then deposit them on quadrant 4. The purpose of doing this is someplace along the line, when you're streaking, you will put down isolated cells which will grow into isolated colonies. In this case here, we're getting that in quadrant 4, putting down isolated cells which grow into isolated colonies. However, if the concentration of the cells had been a little more dilute, you would get isolated colonies on quadrant 3. And if it would be even more dilute, the original starting cells, than that, you would get the isolated cells being put down in quadrant 2, and you'd see the isolated colonies in quadrant 2. And if the bacteria culture you're starting with is extremely dilute, you could actually get isolated cells put down on quadrant 1 and get your isolated colonies on quadrant 1. If you get isolated colonies on quadrant 1, what are you likely to get in quadrant 4? Come on, somebody's going to have to guess.
Uh, no colonies.
Correct, because there will be too few colonies. When you go back into quadrant 1, you might get a colony or two in quadrant 2, but you certainly aren't going to get many. And then quadrant 3 and 4 will likely be just totally empty. Anyways, this isolated colony is not a pure culture yet. So what you want to do is take from an isolated colony here and then streak it on a fresh plate for colony isolations. And take an isolated colony on your second plate and then set up your pure culture. If you were to set up your pure culture from these isolated colonies here, I'm going to warn you about 30% of them will be mixed cultures because 30% of these colonies are formed by more than one cell. And if it takes two cells to form the colony, then that colony, there's a chance that that colony will not be pure. And it's between 0 and 50%. I'm not sure what it is, but the point is that 30% of these colonies may not be pure, and so you should not set up your isolated colonies from the first streak of your colonies because your chance that you have a pure culture is only around 70%, and that's not good enough in microbiology. Any question about that? All right, incubation. So the culture media is normally put in an incubator in order to grow. Incubators provide a physical environment for the culture to grow. You can have a complex incubator like this one here, which can control how much oxygen the cells are going to be exposed to, how much CO2 the cells are going to be exposed to, the temperature the cells be exposed to, the humidity, and then the time. The time will depend on how long you leave the container in the incubator to grow. So the time is dependent upon you. In our microbiology lab, we don't have any complex incubators like this that control the amount of oxygen and the CO2. But we do have incubators that control the temperature. They are usually set at 30 degrees, 35 degrees, or 37 degrees. The humidity, you see that tray there? We pour water in there, and that makes the environment humid inside the incubator. And that's important for growing the plates because if you have a plate of media in a humidified incubator, meaning one that has water in it, the plate can survive for 2 weeks or more before the plate will grow out dry. And once the plate dries, the bacteria will start to die. Like E. coli, if the plate's starting to dry, the bacteria are dead probably, if not starting to die. However, if you do not have a humidified incubator, the plate will grow at most 1 week, and then the plate will dry and the bacteria will die. So we need to put water in the incubator so that our plates can last for 2 weeks or longer in the incubator. And all you do is you put water in there, and it's the humid environment is because the incubator is warm and that makes the air humid. All right, any question about any of that? Obviously, to get the correct temperature as well as the oxygen and the carbon dioxide, you need to shut the inner door, which I don't think you can see that there, but there's a plastic or glass door there, and then that's the outside door, the one you can't see through, the white one. And then once you have your growth on the plate, you then inspect the plate to see colonies. And there are obvious visible colony morphological variations which can help you begin to differentiate different species. You could also determine the appearance of the colonies, the variety of descriptors, the size, the shape, the margin, the color, the elevation. You refer to Lab 1 for a review of the appropriate terms, but you cannot identify the species just by looking at the colonies and looking at the growth. So you then have to do identification to identify the species, and that is done by biochemical tests, at least in most laboratories. It is possible you could identify the species just by sequencing the DNA to the species, but most labs actually run the tests, the biological tests. All right, here's some terms for this lab. You should know the terms. You can skip the references. In this lab, everything we do will be done online, but you are required to understand this lab and then understand the videos that we give you for performing this lab. Like we have a video here on inoculation of an agar plate to obtain isolated colonies, and that's called the streak plate culture technique. On this video, you only need to watch it until 4 minutes and 15 minutes. Beyond that, you don't need to watch it. And you should note that this person who streaked out the plate only streaked out 3 sectors. They did not streak out 4 sectors or 4 quadrants, and either way is fine. And then we have a little video on beta hemolysis, a little video on alpha hemolysis, and a little video on gamma hemolysis shown on blood agar plates. And then we have a little video to show you and talk about McConkey's agar. On this video, you only need to watch it for 3 minutes and 50 seconds into the video. After that, you do not need to watch the McConkey's agar. You should also watch the video we have on mannitol salt agar. Any question about any of that? If not, let me go to the worksheet. The worksheet to Lab 5, answer these questions. Describe a natural circumstance where Staphylococcus species of bacteria might benefit from tolerating salt, meaning salt tolerance. And you cannot say putting it on a MSA plate because that is not a natural circumstance. Any question about any of that? All right. 2, how might hemolysis benefit a hemolytic organism? Meaning if we have a bacteria that can lyse or hemolyze red blood cells, how might that benefit the bacteria? And 3, imagine you have a culture media containing a single bacteria species. This culture may be one of several possible species. Describe a scenario where you use mannitol salt agar, blood agar, and EMB plates to help identify the unknown bacterial species. You must name 2 properties from each plate, mannitol salt agar, blood agar, and EMB, that can help you identify the species. Any question about 3? So you need to describe 6 properties to this number 3. All right, 4. What is the main goal of streaking for colony isolation that was discussed in this lab? 5. What happens to the growth on the plate if you do everything correctly, but you forget to sterilize the loop in between sectors when streaking for colony isolation? In your answer, you must be specific and discuss the growth on each sector. I need to know the growth on each sector to understand if you understand this question, and your answer tells me the correct answer. Meaning if you just state something like, you may not get isolated colonies, that's going to be wrong. Because you need to discuss the growth on each sector and telling me what the growth will look like if you forget to sterilize the loop in between sectors when streaking for colony isolation. 6 is a little similar, but a little different. And what happens to the growth on the plate if you do everything correctly, but you transfer from the stock culture to inoculate each of the sectors? Once again, you must be specific and discuss the growth on each sector. Any questions about 5 or 6? And note in both 5 and 6, you're doing everything correctly except on 5, you're not sterilizing the loop in between sectors, meaning in between sectors 1 and 2. So you're sterilizing the loop before sector 1. And then 6, what happens to the growth on the plate if you do everything correctly, but you're obtaining cells from the stock culture to inoculate each of the sectors. So obviously on 6, you are sterilizing the loop in between sectors. All right, 7. Suppose you successfully streak a plate and obtain isolated colonies of 2 different bacterial species. That is, you started from a mixed culture. How can you use this plate to create a new pure culture of only one of the bacteria? You must be specific in your answer, going through all of the details you need to get an isolated pure culture. 8. In the table below are a variety of agar plate media available to microbiologists to cultivate bacteria. Denote whether each medium is non-selective, a general purpose media, selective, a selective media, and or a differential media by placing an X in the appropriate column. You may use the table media in the lab module to answer these questions. So nutrient agar, if it's non-selective, put an X there. If it's selective, put an X there. If it's differential media, put an X there. And I will advise you that some of them have more than one X. Actually, some of them have 2 X's in it. And I'll give you a hint. There is never a media that is both non-selective and selective. That is impossible. And let me see if I can show you something. If you were to come into the lab, I would show you a book about this size, about this thick. Hopefully you can see this in front of my face. And it's actually a little bit longer, as well as a little wider than this. So just a little bit bigger than this. On each page of this book, we have one media. So that tells you all the different media we have in microbiology for a microbiologist to use, because each page has a separate media. And you can tell there's a whole lot of pages in that book. And then 9, which agar plate or plates were inoculated without using streak plate technique, meaning without using streaking for colony isolation? Plate A, plate B, plate C. And that's it. Any questions about this lab? All right, I'll be here until 4 o'clock, or the last student logs out, to help you with any questions. So go ahead and get started on the lab.